Hi, my name's Natalie, and today I'm going to be talking about measurement of frontal midline theta oscillations using OPM-MEG. So human electrophysiology is dominated by oscillations, and these oscillations occur across a wide range of frequencies, ranging from the very slow, which we call delta under 4 Hz, all the way up to the fastest, which we call gamma over 30 Hz. And these oscillations relate to core processes, things like cognition and connectivity. But today we're going to be focusing on the theta band, which is from 4 to 8 Hz, so relatively low frequency. And so theta is a marker of cognition. We see it in working memory tasks, which is what we're going to be talking about later, but it also has some other interesting relevance. So in early life, in young childhood, we see that the dominant oscillatory frequency is actually within that theta band. So you can see here from ages 0 to 5, the peak frequency is within that 4 to 8 Hz range. And then it speeds up to over 8 Hz and closer to 10 Hz through later childhood and into adulthood. And then on the other end of the spectrum, in atypical aging, such as dementia, we see a significant power increase across the theta band relative to healthy aging. And so it's really important that we're able to measure these low frequency, these 4 to 8 Hz oscillations. So how are we trying to measure them? We're using magnetoencephalography, or MEG. So MEG is measuring the tiny magnetic fields around the scalp that are produced by currents from the neurons within the brain. And so this technology has been around for a very long time, and they typically use cryogenically cooled sensors. And so here we've got a picture of a CTF system, a cryogenic MEG. And what we have is this big dewar of liquid helium that's holding all of these sensors that are very sensitive to these tiny magnetic field changes. But because they have to be kept cryogenically cooled, the sensors must be kept further away from the scalp and held in this rigid one-size-fits-all helmet. And participants have to remain perfectly still. So this has a few problems. Firstly, the amplitude of the signal decays with the square of distance. And so because we have to have these sensors slightly further away to keep the cryogenic liquid helium away from the scalp surface, we're actually measuring a lower amplitude signal than if we could put those sensors closer to the head. And then also our participants have to remain perfectly still. So it's very challenging for certain participant groups. And so recently there's been developments in these sensors called optically pumped magnetometers. And these operate at much closer to body temperature. They're also really lightweight and flexible. You can see a picture here just held in a hand. And so we can put these in helmets and wear them directly on the head. And this obviously has a few advantages. Firstly, because we're able to bring them closer to the scalp surface because they're not cryogenically cooled, we're able to pick up that higher amplitude signal. Secondly, because we can put them in these helmets and wear them, we're able to allow some natural movement. And also they're flexible and lightweight. We can move them between different helmet sizes, and so we can size match these helmets. So OPM-MEG at Nottingham has been developed since around 2017 when we had our first one-channel recording. And then a wearable array was developed by 2018, and we started including this movement element. And then we went from these personalized 3D printed head casts to these adapted bike helmets to be able to measure signals from children by simply drilling holes into a bike helmet from Amazon and placing these sensors in there to get these OPM sensors close to the scalp to pick up these signals. Then we started getting whole head recordings, and towards where we are today, where we have 174 channels in these rigid 3D printed helmets that we have size matched. So here we've got a child in a 4-year-old helmet, and we have various different sizes to match different head shapes and sizes and get those sensors as close to the scalp surface as possible. However, why is it a challenge to measure things like the theta band with OPMs? Well, the hardest thing to measure with an OPM is low frequency. This is mainly for 2 reasons. The noise floor in the low frequencies is higher in OPMs. They have this 1/F noise profile. And also, the sensors are magnetometers, and so they measure a change in magnetic field. And so if we move through a magnetic field, we will measure the change in magnetic field due to the environment. And this can be much larger than the brain signals that we're interested in and can drown out what we're actually trying to measure. And so therefore, we have to control our magnetic field environment. We have to keep a very low magnetic field environment to allow these movements without losing those brain signals. And so we use active magnetic field nulling, and we use a magnetically shielded environment to be able to suppress the magnetic fields from Earth's magnetic field down to the nanotesla and even picotesla level. And so we wanted to test our OPM-MEG system with a cryogenic classic MEG system. So we scanned 14 healthy participants, and they underwent the same working memory task in our conventional cryogenic MEG system and our OPM-MEG system. And so we used a working memory task. Like I mentioned, we see this theta-band oscillations increase during working memory. And so what we used was something called a n-back task, specifically a 2-back task, where we had letters presented on a screen and participants were asked to press a button if the letter on the screen matched not the previous one, but the one before, so 2 previous. So it's actually quite a challenging working memory task because the participants have to hold multiple letters in their mind, and then they have to press a button if it's the correct letter from as 2 previous. And then we also had this rest period. So we did 20 seconds of this activity and then 20 seconds rest and repeated it. And then we also took anatomical MRI scans to be able to co-register our MEG data to each individual's anatomy. And so here's some of the results. What we did was we used beamforming to work out where in the brain that signal was most likely to come from. So we used a pseudo-T statistical approach where we contrasted the theta band power specifically. So we filtered between that 4 to 8 hertz. We contrast that during the task window, so while they're doing this working memory task, with the rest window. And in both our cryogenic MEG and our OPM-MEG, we saw activation in the frontal cortex. As expected, this frontal midline theta oscillations, which are typically induced with these working memory tasks. And we see a very similar spatial map for both systems. And then also we did this virtual electrode approach where we basically took the peak of those maps and said, if we put an electrode here, what would the signal look like? So here is a group average and trial average of what we'd expect the signal to look like from those peak locations. And we see for the OPM and the cryo-MEG that these are very similar signals. And the signal to noise for these were both on the order of 5, which is great. And then what we decided to do was go, okay, if we're measuring the exact same signals, we should see those who have a strong theta response in the cryogenic MEG should also have a strong theta response in the OPM-MEG. And so we compared on an individual level and fit this linear response to it and confirmed that the individual signal to noise is preserved between our cryogenic MEG signal and our OPM-MEG signal. So we are preserving measuring these same signals between the 2 MEG systems. So just to summarize this part, despite allowing movement, we're able to obtain reliable responses in the theta band range. And our OPM-MEG system can measure these frontal theta oscillations with a comparable signal to noise as cryogenic MEG. And individual temporal and spatial signatures are preserved between these MEG instrumentation, which is a real confirmation of OPM-MEG measuring the same signals as our cryogenic MEG. And so why is this important? What do we want to use it for? Well, now we can focus on developing a paediatric OPM-MEG. So we have some initial results from a sensory motor task with 27 children across quite a wide age range from 2 to 13 years old. And so we have these braille stimulators where we have small pins that tap on the index and little finger of the children on their right hand while they're watching TV. And we see this expected event-related desynchronization across the beta band, which is localizes to the contralateral sensory motor cortex, exactly how we expect, but these signals are in children too. And if you'd like to see more details on this, you can go to my poster number 1246. So just to conclude, low frequency activity is the biggest challenge for a wearable MEG because of this high inherent sensor noise and the movement artifact. But we've proved that we can measure theta-band oscillations using our wearable OPM-MEG and have a comparable signal to noise to conventional MEG. And it demonstrates we can obtain high quality data in these frequencies that are critical for children, and suggests that OPM-MEG could also be used to study atypical aging too. So this work was not completed alone. I'd like to acknowledge and thank my colleagues at the University of Nottingham, QSpin, who have developed these incredible sensors that allow us to do this work, our collaborators at SQUID-based for helping us towards these paediatric OPM-MEG studies, and the spin-out company Circa for commercializing this system and working towards developing it into something bigger and better. Thank you so much.